Okay, well, welcome everybody to Genomics in the Cloud Book Club. This is our first week, and I'm your host, KT Picard. I wanted to give some background about our meeting today. So our agenda is a welcome and some just basics about the book club. A little bit about our weekly schedule, some time for everyone to talk about themselves and their interests, and then anything else that we'd like to talk about on the call as well. If there's other things you'd like to cover during this call, I'll stop here. Great. I'll pause here. In fact, I'll pause and make sure that Mikhail can hear us. Can you hear us okay?
Yes, I can hear you well.
Great, fantastic. And welcome. It's good to see you on the video. You can see on the video too.
Yeah. So
welcome. So it's good morning, good afternoon, good evening. We have about 15 people, as I mentioned, in the club. In 9 different time zones. So this ends up being kind of a reasonable one, especially if you're located in Asia. But it's always challenging when you're doing these things globally. So we'll be walking through Genomics in the Cloud. This is our book for the next few months. This is by Geraldine van der Arwera, there we go, and Brian O'Connor, published by O'Reilly Media. The book is available on Amazon or at the O'Reilly website, or as Richard has pointed out, depending on where you live, your library may cover it, and you may be able to look at the book online. I think everybody here is on Slack, so there's the link to the Slack channel, and that's where we'll put announcements about the meetings and also where we'll share things, questions, and things we've learned. The idea is to do weekly meetings for the book, basically a chapter a month, or sorry, chapter a week. And that takes us through roughly March of next year. We'll adjust as the holidays come through, and if things get a little more challenging, especially when we get into some of the GITC best practices, if we need to take extra time, we will. I should have said at the beginning that this meeting is being recorded, and those recordings will be posting onto a YouTube channel, and the link is there. Eventually, we'll get a more tidy link, but there it is. In case you miss the meeting, you'll have a chance to catch up. All of us are on Twitter, so you can follow. I've created a list of the Genomics in the Cloud Book Club members and friends. The friends include one of the authors. So Geraldine actually is in our group. She's also on Slack. So you can follow. I've actually found it very interesting to learn about what everybody's interested in. And so that list, if you haven't subscribed to it, gives you a good sense of what everybody's up to. And just for fun, a little bit about our mascot. You see him down at the bottom right, and he's our number counter, our slide counter. He is the milk-spotted pufferfish. He has been sequenced, and there's a link to it there. And by the way, I'll post these slides to Slack when we're finished. And the commonality is that, like the pufferfish, cloudburst computing can get bigger on demand, and there's the fun bit there. Anyways, it's a fun mascot, and there's more in the book about him if you're interested. So I'll pause there. Any questions or suggestions, comments?
KT, I don't know if you're going to talk about it, but I'm sort of curious why you picked this book and not something else. And
oh, sure. So there's another book actually that kind of competes with it called Genomics on AWS. This one in particular I liked because it really delved into GITC best practices and had a lot of workflow automation. So those two areas. I didn't know it had a chapter on Jupyter, which we'll get into as well. But I like the overall approach to it, specifically taking those software tools and putting them into a Docker and using Docker to complete the pipeline. So that was the main reason that I chose this book. We'll be using the Google Cloud Platform, which there'll be free credits to use along the way. Personally, I've done most of my bioinformatics work on Amazon, so this will be new for me, which I'm always game for trying something new. But yeah, that's kind of why.
Mm-hmm.
And suggestions are welcome. I mean, if there are other areas we can, if there are other books that you like that have helped out, feel free to mention them, and we can use, reference some of those materials as we go along. But there's a nice structure to the book. It has a nice flow in terms of. In fact, we'll go to that next. So it might be a nice segue. Any other? Was that responsive enough? Did I get at it?
Yeah, no, that's good. I just sort of, I don't know a whole lot about this subject, and I'm just curious why. If you had known Geraldine beforehand, or if this is a well-regarded book that's used in lots of places, or if, because like you said, I'm more familiar with AWS, and I just don't know enough about genomics and about, cloud genomics to know if maybe all the cool people are using Google for this stuff and not AWS, and maybe it's like a better platform for this sort of thing, and because I know AWS has its own genomics components, but I don't know enough to be able to say if it's better or worse or anything, and I was just sort of curious if there was more like a backstory or something that like I said, maybe the cool people are using this one, and I just need to know more about it. I'm always up for learning new things anyway, but. Yeah,
no, thanks. And I the same. I because I've been doing most of my work on AWS. This will be new. And again, I just because the book was laid out this way, I have no ties to the author at all, and no other than I saw the book on Twitter. I think it was published back in April of 2020. And my mother gave me the book for my birthday in October. So that's kind of that's how I got here.
So did Geraldine just kind of show up after you announced that you were doing this, or
yes,
you have some other kind of connection?
So it was really kind of cool. So we'll get into that in a few slides in terms of how this all came about. But basically, I wrote to her and I said, "We're doing this book club," and she said, "That's great. We've been wanting to do something like this. Would you be willing to host it?" And I said, "Sure." And that's how this all came to be. So, and I didn't ask her to join. Actually, she said, "Could I join your Slack?" And I said, "Absolutely." And you've seen she's chimed in on a number of occasions. And so I really appreciate the involvement. But no, other than that, there's no ties or interests or anything of the sort. This is a completely.
No, I mean, I really appreciate you putting this together. It's kind of cool.
Yeah, thanks. And I got inspired. And in fact, I think, is that our next slide? Yeah, this is kind of our next slide. If you jump down to this R for data science, this is how I bumped into it.
Yeah, that's a great book.
There's my wife was studying it. Emily Whistle is a grad student. I think she's at Emory. In any event, she was doing a book club on Twitter, and she's learning advanced R. And so I got in touch with her and I said, hey, can you give me some ideas on how you've structured this, what you've learned? And so she pointed me to the whole R for data science community. And it turns out that they have a Slack channel, which I joined and largely copied the format that you see here on the channels and used that idea. They actually have a nice community going with a website and the whole.
Yeah, and part of it, it's very good. And they're very responsive too when you have questions. Yeah.
Yeah, exactly. So we're probably not quite as large as R in terms of, it's a small, bioinformatics is a very small world. Nevertheless, it was inspirational, and you'll see if you've been on that R for data science Slack channel that there's a lot of similarities. Any other comments or thoughts? So on the schedule, as I mentioned, we'll do one chapter per week. It starts off a little slow, and I really encourage everyone to participate regardless of your background, because we'll learn so much from people that are on the call. So for example, next week is just kind of a basic, why are we here and what are the things that we'll cover in the book. Chapter 1 gets into, there's a chapter on genomics, a chapter on cloud computing. There's chapters on the basic nuts and bolts of genomics and bioinformatics. So one of the things I liked about the book is that it doesn't assume that if you're coming from a computer or IT background, it doesn't assume a heavy background in genomics. And conversely, if you have a heavy background in genomics, it doesn't assume that you have a heavy background in cloud computing. And that's one of the things that I like about this field, is that it takes, a lot of disparate knowledge of lots of disparate fields to kind of pull together. And so again, it's nice that they take the time. So there are 3 chapters there that are roughly grouped into intro. And then we start to get into the heavy lifting of setting up Google and running GITC, and then the workflow and so on. So, excuse me, you can see on the slide that the channel list. And by the way, is the channel list covered up by the chat, or can you see it? I've got a chat window up. Does it block anything?
Oh, I can see it fine.
I see the whole screen.
Fine? Okay, great, cool. I'm still, it's been a while since I've done Slack. So about Slack itself, I think everyone's figured out that the GITC Genomics in the Cloud announce channel is the general channel on Slack. We use it for general announcements. As I mentioned, the chapters are grouped by section. So sometimes you'll see multiple chapters that are grouped together. So I think there's like 3 chapters for GITK. So those are grouped together, and so on throughout the book. So you can see that there's roughly 7 areas that we'll cover. Lastly, at the bottom, you'll see wins and feedback. And I, again, stole this straight up from the folks on the R for Data Science. And it's a fun place where if you figured out something like, oh, it was really, I had to do X or Y to get the GCP, the Google Cloud Platform, to behave. You could share that with the team, or I got married, or whatever you want to share with the team. It would be nice to develop community that way. So I wanted to just kind of cover again the thanks to the R for Data Science folks, Emily for the inspiration, and Geraldine for the support. It's been a good journey so far. So good journey. So we've got time. So I was hoping to walk around here. I'll turn off my sharing and walk through introductions a little bit, let everybody get to know each other. We've all got video. That's great. There are 5 of us, so 2 or 3 minutes to let us know about your interest in genomics or cloud computing, or if you've done book clubs before, that would be really helpful. This is my first book club. Certainly online, and in the age of COVID, it actually kind of makes sense, I think, almost. But yeah, either I can kick it off, or if there's somebody who wants to jump in,
I'll start. So
great, thanks.
Katie, and you and I guess have known each other for the last couple of years, I guess.
Yeah.
And I remember you as a old Symbolics Lisp user, so we have something in common from like that.
Yeah.
I think that's goes back a few generations there. And otherwise, I'm not an expert in genomics or anything else, but I consider myself to be an amateur at this stuff, but I've just been really fascinated by it. So for the last, I don't know, probably 10 years, I've been dabbling in various different levels of trying to understand more about genomics. My background is in software engineering. These days I use mostly R. My claim to fame, I guess, from genomics is that maybe 7 or 8 years ago, I got really interested in the microbiome, and I did a lot of work on studying mostly 16S, taxonomic results, and I've written a lot of software and a lot of analysis for that, mostly for my own microbiome, and worked for a while at the San Francisco startup uBiome, where my job title was citizen scientist in residence. I was mostly just taking what I'd learned about the microbiome and how to do your own self-analysis, and spreading that to as many people as would listen to me. In terms of my interest in genomics in the cloud, again, it's just overall interested in this space. I thought it'd be fun to be part of another group of people who are doing the same thing, and that's why I'm here. So I live in Seattle. Used to travel to San Francisco Bay Area a lot, but with COVID, I'm kind of stuck here in my little green screen room. Lived for a long time in Asia, so that's a little bit of my background as well.
Fantastic, Richard.
Nice to meet all of you.
Let me ask you, so Yeah, thanks. We've talked about Google Cloud Platform before. Have you used Amazon in the past?
Well, I use AWS for a company. My business partner and I are using it for a company we call personalscience.com. And it's mostly just for hosting, and I do various different things with like Lambda instances and a couple of things like that, but nothing serious and nothing at all related to genomics other than just as a place to to store my microbiome data. Right.
Well, the good news is I think you'll find the movement of microbiome data to human genomics a fairly easy lift. Yeah, lift and I don't know,
right.
a fairly easy drop in. So yeah, yeah, great, thanks. Shall we just go across, on my screen? Matt, you're up next. Would that be okay?
Yeah, sure, that's fine. Thanks for the introduction, Richard. So I'm, I guess, coming more from a biological background. I actually started off as a marine biologist, slowly transitioned into sort of more of the bioinformatics. I did a master's in basically working with corals, sequencing, did a lot of RNA-seq. The project, I was doing genome assembly actually for that, or transcriptome assembly, I should say. And we had problems with the pipeline. It wasn't a full build to put on NCBI. We weren't quite ready. The cloud computing, so I'm from Canada. We typically use what's called Compute Canada, a large shared compute cluster. That had kind of ran out. I didn't have access to that anymore. So I had, and then new methods had come out around the time I had done my defense. So I had an opportunity to kind of revisit the data, but I wanted, or I was able to use AWS, and this is like 2016. So I never managed to get Lambda instances running. I tried that recently, so I'll have to speak with Richard about that. But I'd kind of dabbled with it a bit. I'm an R programmer primarily, kind of getting more into Python now. This is my first book club here, so kind of COVID's pushing me towards it. I had heard about the book and talked with Geraldine a couple times on Twitter. She's super nice about it. We're actually, I'm working with rare diseases right now, so we're concerned about the cloud, I guess, for safety, privacy concerns and ethical concerns. So I'm really here to see where this is going. Just to give you a little bit of background of where I've seen the cloud being used recently, I've been working with a genomics browser called JBrowse 2, which was just released from beta. And so the reference genomes, you can't use your own locally. They need to be stored in S3 buckets currently. Another tool, Hail, is used for working with large scale genomics like GWAS results. And it's been used by the Nomad group, which has like 141,000 exomes and whole genome sequencing. They're doing all their work. They were linking with GITC, which is a large biobank of tissues, and basically getting annotations from that could be used to add information to annotation. And so to run their software, you need to be able to run it in Google Cloud Platform or on AWS. So kind of seeing that it's moving towards the cloud. We're not quite there yet. So I'm really interested in the book in that perspective. I'm from Canada, from Calgary. So in my free time, I'm a little bit interested in backcountry snowboarding. I went the other day. There's a lot of snow here. It's probably still sunny and warm in California. So that's my intro, and looking forward to hearing from the rest of you guys. Thanks.
Thanks, Matt. I caught the sweater, so I figured I have to put names together with faces, and so yeah, I'm assuming it's a little cooler there than it is. I have to say though, it's 0 Celsius this morning, which is for San Francisco in the area, it's a little chilly. But anyway, thanks for all that. We'll circle back to you mentioned privacy and ethics. We'll circle back to that because I think it's an important one. And in my day job, I work at Philips Healthcare, and it's one that I deal with a lot, especially with in the cloud and the issues around de-identification and that sort of thing. So thanks for bringing that up. We'll be sure to circle that into the conversation.
Wonderful, and I look forward to hearing your experience with that as well too.
Sure, great. Yoris, I'm assuming that's a Yoris. Am I getting that right?
Yep, that's absolutely right. I was going to say, I'm dialing in from Cambridge, so I'm sort of climate-wise in between Calgary and San Francisco, but it's way warmer than 0 degrees here. So.
Good enough.
Yeah, so my background is, I'm not a biologist. I'm an applied mathematician by training and sort of a software developer by trade, and drifted into life sciences because I work for a consulting company. And we did actually a lot of data analysis for also microbiome analysis, mostly 16S, metagenome sequencing. So I'm really curious to sort of compare and contrast with human genomes and other kinds of trade-offs. And yeah, the reason why I got interested in the book and sort of the book club as a whole is because we sort of ran a little bit into the problems that the book discusses in terms of reproducibility, compute at scale, cloud computing, and sort of being able to trade in money for increased throughput, so to speak. So yeah, I'm interested to sort of see how the techniques that the book discusses will match with my experience and how that will broaden my understanding. And also, yeah, I'm interested to both learn a new subject area and yeah, hear from you guys and your experiences and things.
Great, thanks, Russ. So when you mentioned 16S, is that in human or in something else?
Bacterial genomes. So it's a small, about 1,000 base pairs gene that you can use for taxonomic analysis. So you can with very little effort answer the question of like who's living in my gut.
Are you looking at but you're looking at human 16S analysis, right? So
yeah, human
like gut microbiome or whatever.
Yeah, exactly.
Great. We'll have a subcommunity for microbiome here. And you mentioned reproducibility, and another topic. There's actually a chapter in the book about this, and I think given we also have this reproducibility. Some people call it a reproducibility crisis in science, and a lot of it I think comes down to really good documentation. So we'll cover next week actually the so-called fair practices. In scientific literature, there's actually a chapter dedicated to it, as I mentioned, on reproducibility. If you're an R person, you kind of have some of the background and understanding how it's kind of self-documenting, so that when you go to reproduce your pipeline, it's kind of understood where you came from. And that's a little less obvious in the world of genomics pipelines. So I think it's actually important that they call that out, and thank you for bringing it up. So we'll get to it. It isn't really till the end of the book, but it is something I'm also very interested in talking about, because when you publish, I think it's terribly important. I've read so many papers where it's just that the methods are, especially when it comes to software methods, either the methods are vague or worse yet, the code's not available. Right, and that's often the case. So I think as good stewards of science, we owe that to our communities to put that in place.
Yeah,
so, thanks.
Absolutely.
Thanks again, Mikhail.
Yes. So
over to you.
Hello, hi everyone. I'm Mikhail, and well, I'm originally from Indonesia, and I'm currently doing my PhD in the Netherlands. And I'm a medical doctor by training, but a bioinformatician by accident. And well, mostly because I think 3 years ago now, when I started doing my PhD, I was originally assigned to do work in the wet lab, but then it didn't work out. And at that time, I would say I was at my wit's end on, well, what can I do to further this PhD? Because I'm on the PhD under a government scholarship, so quitting is not an option, not at all. So yeah, so then I came across at that time RNA-seq data analysis, and I dabbled in that. But then after quite some time, I noticed that, well, I do know a lot about RNA-seq data analysis, but then I don't know anything at all about the rest of the genomics field because I was really well studying what I needed. And as I told you before, I was studying RNA-seq because of desperation and accident. But then I came to enjoy it a lot. So I now really look forward to learn about the rest of the genomics as well. Right now, currently, I'm working on a single cell genomics project under the Human Cell Atlas project.
Mm-hmm.
But I haven't really tried, well, I have tried going through the first chapter of the Genomics in the Cloud book, and I was really amazed by how it provided guidance to navigate the Google Cloud platform, because I have tried using the Google Cloud platform, but there are just so many options, and I was really overwhelmed, and I can't even start anything because I don't know what to click. And I think there was, I think Matthew mentioned about the future of cloud computing, and I'm just really interested in that, also in genomics. So, I mean, this is the best. This is two things that I like combined together. So why not? And yeah, so this is my third book club actually. So I'm really happy that I'm not sure why, but it's just so much easier to find study groups for things like this, for bioinformatics or programming or statistics, compared to study group for wet lab. Because for wet lab, it's like no one really knows, kind of like no one really knows what they're doing, because they just putting random amount of liquids and try which one works out. Well, that's what my experience is. But then people don't really share what works and what don't. But it's just completely different with bioinformatics. People just document everything, what works, what don't. And it really helps in my learning, because I know, like from internet, without internet, I don't think I can, get through the desperation phase. And yeah, so I owe a lot I owe a lot to the community for, for such study groups and for a lot of freely available resources, really.
Right. Great. Well, thank you for that. And thanks for being with us. You come to the right place when it comes to figuring out the intricacies of GCP. We'll stumble through this together, right? And it'll be new for me. Again, I think all of us are coming from different places and listening to everyone. It's kind of 50-50. Some people are coming at this from a computer science background. Others, like yourself, are coming to this from either a medical or a genomics background. I'm curious, Mikhail, your MD, is it in a particular field, or are you a general practitioner?
I'm a general practitioner.
Okay, fantastic.
Yes, yeah, previously I actually planned after doing PhD to specialize, but then I guess I really like bioinformatics a lot.
And are you thinking about it in terms of, in terms of an area of rare disease or cancer, or any thoughts there?
Right now I'm doing the single cell analysis in the context of immunology as well as infectious diseases.
Okay, infectious. Okay, good.
Yeah.
Good, thank you. Okay, great, I missed that. Okay, well, a little about me. I don't know, curious by nature. By the way, my dog, if you hear him, is snoring in the background, so that's what that is. Richard mentioned the old link to, or a link to symbolics and Lisp, and that was a nod to my days at a company called Thinking Machines some 30 years ago. I met my wife there in Cambridge, in Massachusetts. And so I came at this also from a software engineering background. We were using supercomputers back then to, this is for BLAST and the beginnings of the Human Genome Project. I knew very little about biology and saw all of this as a giant computer science problem. Took a 20-year detour into medical imaging, and that's where I've spent my career. about 10 years ago, I went back and got an MBA and focused on direct-to-consumer genomics, realizing that the kind of golden age was starting to dawn, with the sequencing of the publishing of the first human genome about now 20 years ago. So the last 10 years have been getting some genomics chops and trying to understand biology and going back to school to do that. I had our family sequenced, and so that's one of the drivers is that I have 2 kids with some special needs and autism, and so part of it was to understand some of the underlying genetics of their autism. So I've done some work there. I publish my stuff on a blog. I'm sure many of you do. I'm at genomedad.com, where I kind of publish my adventures in bioinformatics, and all the stuff that I've done on AWS is documented there. I'll post on the chat window. This is back to Matt's comment about JBrowse and having to have things on an S3 bucket. So you'll find on that website, my genome is hosted there on an S3 bucket. It has I'm drawing a blank. It has a web server in front of it that allows you to get to it. It's not the fastest thing in the world, I think, to put a T1 micro or T2 micro, whatever it is, instance in front of it. But anyway, it's all there. It's CC0, meaning that it's fully in the public domain. I've signed a HIPAA waiver. If you're in the States, you have to, whether or not you can let other people use it. Anyway, it's fully, completely available. So if we do need to muck around with a genome, there's a spot, a place for us to play with. Let's see, I think that's about it. And my interest in this, of course, is I've never played with Docker before, and I've never run a pipeline from soup to nuts. Part of my motivation for this is that when my wife and I had our genomes done, the genomes were done on a different set of bioinformatics tools. We crowdsourced the funding of our kids' genomes, and that went through kind of a more traditional GITC pipeline. And so part of my goal from this whole book is to put all of our genomes through the same pipeline. This kind of gets to the comment about reproducibility, so that we can have something that's normalized and makes more sense. So along the way, you'd mentioned Mikhail, you mentioned documentation. We'll get to Jupyter notebooks, which I think is just a super cool way to document and share information. Has anyone worked with Jupyter notebooks before? Is that new to everybody? It's new to me. Got a couple? Cool. Okay, so this is what I like is we got a great mix. There's some obviously really some strong brain power here on this call, and again, everybody's coming at this from a different place. So just know that the parts that you're a little concerned about or don't have as much experience with, there's basically somebody on the call who does. I'm absolutely convinced that if we could put our 5 brains together, or the 15 brains together, this would be an amazing hive to get things done. So, okay. Any other thoughts there? Thank you. Those were great intros. Really appreciate it. Okay, let me bring the slides back up. I think we're about there. So this is the thanks for joining us today. We'll start again next Monday, same time. This seems to work out fairly well. As I mentioned, we'll cover chapter 1 in the book. It is a relatively simple introduction to what we'll be learning, but we'll also use that time to lay out more detail about the structure of the book itself, and an overall view of how we'll put all this together. So bear with us the first 2 or 3 weeks. Again, some parts will be more interesting to you, other parts less so. But once we get into the GITC stuff, and the cloud computing bit, that's really where I think the heavy lifting will come, and we'll really get into it. But so, like I say, a couple of weeks of preliminaries to get us all kind of level set, both with bioinformatics, with genomics, with cloud computing, and to really just kind of, this is the space to explore those questions that you haven't been able to ask somewhere else. That's my hope. Along the way, if you have questions, I encourage you to bring up, let's see if I got Slack here, yeah. Bring up, I gotta share again, one second. Am I sharing?
Yes, I do actually. So,
yep.
So how's the format of the book club going to be? I mean, is someone, or maybe you, is going to be the sole presenter of for the whole book, or are we going to, I don't know, make a Google Docs and then everyone just fill in? So for which chapter someone's going to present, or how will it be?
Yeah, thanks for that, Mikhail. So I'm open. I didn't want to burden everybody with this, but in the absence of I'm certainly with my kids, I call it voluntelling, but I don't have that expectation. If as we go along, if someone would like to pick up a chapter, like if genomics is your thing and you'd like to pick up that chapter, cool. That would be great. If your background is computer science and you want to pick up, there's a chapter on basic concepts around computing, and that's your thing, that would be cool as well. If there are no volunteers, I'll put together some slides. So I'd welcome the kind of, especially in these early chapters, because clearly there are those people on the call who have much deeper expertise than I do in various areas. So yeah, let's mix it up and let's use the chat to on Slack to kick that around. So if there's a chapter that interests you and you'd like to present, by all means, please let us know. I would welcome that, actually. Okay, so I'm sharing the Slack channel, and so this would be, again, just to quickly walk through this, announcements are here. I think everybody's got the hang of it. The intros, everybody's had a chance to write a little bit about themselves, so thank you there. The intro channel then covers the basics. If you have a question, if you hit the I button for information about this, there's always a description about what it's for. So this is ask questions for chapters 1 through 3, and similarly for the rest of it. It asks questions about in the next one, chapter 4, first steps in the cloud. So you'll see that these numbers don't correspond directly with the chapters in the book, but they are grouped by just kind of sections that make sense, I think. So there's a number of chapters, for example, on GITC, and so on. So that's how the Slack channels are put together. I set it up so that everybody that's in the book club has full access to all the channels. Are already signed up for them, so that everybody will be able to see what it is that you post. Great. Other questions, thoughts before we wrap up? So I'll post the slides on the announce channel, and I'll tag them appropriately so that if you miss a week, you can search for slides and find what happened on that during the week before. I think we're in a good spot, so I'll stop sharing here. The links, I'll also put the links that were in the PowerPoint. I'll put those on Slack as well. I'm assuming everybody has access to PowerPoint. If not, I'll post the slides both as PowerPoint and as a PDF, and that should be universal enough that you can get some viewer or something to access the slides. And oh, right, okay, yeah. So I think that's about all for today. I'll pause one more time. Any other questions or thoughts before we call it a day?
Nothing special. I guess I have a couple other additional kind of random things that'll probably occur to me that I'll just post in Slack. And I guess one of the things I encourage people is that if you see something in Slack, it's really helpful if we know that you actually read it and that you care. So if you care about something, please make sure that you like it or whatever in Slack. Just because I'll post a ton of stuff if I know people care. So and I know that
we got thumbs up. Yeah.
I've got a ton of other Slack channels that I follow as well. And you know, I'm always interested in hearing pointers for those of you who, you know, have run into other interesting Slack channels or other books or other, you know, other resources. It's always fun to, you know, fun to learn about more of that stuff. So looking forward to hearing more from you guys as well.
Yeah, thanks for that, Richard. So same, if there's a book that you see, that, for example, when we delve into the genomics section, if there's a book that you found particularly helpful in, you know, covering genomics, feel free to, you know, put the title out there. Similarly for cloud computing, or for when we get to Docker, I have a number of resources, so we can supplement that way. And again, trying to make this as enjoyable as possible. But yeah, to your point, we had a nice little discussion about Dante Labs for sequencing the other day, Richard, and Geraldine popped in too. So I was really pleased with that. It's really nice to see the involvement. I'll do my best. You know, we either are. My sense is we're either in school or have day jobs as well. So I also am fully aware that this is on top of everything else. So thanks again for being part of this, and for making this, for me, something really exciting, and I think something we'll all benefit from. So thanks again for putting this together.
It's been great.
Yes, thank you very much.
Yeah, I really enjoyed it.
Yeah.
Okay.
Thank you for
all right, everybody. We'll talk next week.
Thanks.
Okay, thanks.
Bye.